For your speech. Thanks, I got it. It's a good plan. Well, there's so much again. No worries. Yeah, that one has it.
That's why I was on the floor. I was like, I just woke up 30 minutes later.
So either of you. I'm very scared. Last week, back.
All right, guys, let's get started. Today I'm not gonna put up a quiz question, you should be fine. Out of everyone's answers from the last quiz, we're gonna do a tie-up day on bringing all the material together on the 7th. That'll be on Wednesday. On Friday, we'll be doing a review where I'm hoping you guys will bring questions. If not, I'll bring questions myself and try to challenge you guys. And then our exam will be on Monday, on the following Monday. I'm still trying to set it up right now. I normally, my exams are, I do like vocabulary in the front, and then it's kind of like matching vocabulary. Then I'll have like a second section where it'll be like small response questions. It'll be hopefully 1 or 2 sentences for you guys. And then in the back, I normally put like the longer, bigger, more challenging questions. If you've taken my Bio 133 course, you probably have an idea of how my exams look. I normally include diagrams, or, you know, I do free response. I give you a lot of space for you to kind of argue with me. If you draw diagrams to explain pathways or signaling, make sure you put a key that goes along with it. So that may give me a little bit of a light of what are the steps or where you're trying to come with the story. Yeah. And then the things I would say from the quizzes is make sure your writing is very clear. And I need you guys to be very, very careful with your RNA. That's one of the things that I was catching early on, right? We're going to be talking about multiple types of RNA in questions to make sure you're bringing out what you're talking about. Another one, if you're like, hey, I don't remember exactly how to spell that word, or I'm kind of close on the name, you can make up for it by giving a little bit of what it does. You know, hey, there's an RNA molecule, I don't remember the exact name, give like it either, for example, for tRNA, right? You can describe it up like, oh, it's part of the, like it works with the ribosomes, and it has a part that helps the mRNA, and it has another part that carries the mRNA. Technically, you didn't write, you know, transfer, so RNA, but at least you told me what it does, it's a big function. Google names for me, I always look at it like, if you're reworking that field and that's your work, you're gonna have Google like yourself, right? So knowing the exact naming, even if you get a little bit off on the naming, I'll be okay as long as I can guess it. If you give me a function, I'll be like, hey, if you understand the material for the importance behind it, I'll let go of the little typos. Yeah. Handwriting, another one from the last time, please write as clear as you can. Avoid like, crossing off big sections or like jumping around too much. If you do, I'm gonna print out the test, so all my questions are on the front, and the back of every sheet is gonna be empty. If you feel like you've written something and you kind of, it's like your brainstorm idea, and now you have like a better way to write it, cross it off and then go on the back page and give me something back. Just tell me what question it belongs to. Yeah. Yeah, so that should be pretty good. We'll talk more once we get closer to it. I think on Friday you'll probably get a little bit better about it. All right, so let's get into today's stuff. Today we're going to be still talking about proteins and how they're folding, and more importantly, the misfolding side. You guys, I hope you guys all did the reading before coming to class. Yeah. Do you guys see how awesome it was? Excited? Yeah. It was beautiful. I was reading, I was like, oh my God, this is freaking gorgeous. I'm like, midterm questions are coming from here. It's gonna happen. It's that good. The mechanisms of control and regulation are just like A1. Like this is like the sweet stuff. I can literally teach the whole class off these sections. That's how good they are. So you know, hint, wink for the midterm. Make sure you do it with a reading for this section. I think it's gonna be my big challenging question. All right.
For this upcoming test?
Yeah,
Okay.
it's gonna be on it for sure.
Good to
If
know.
I don't do it, then I'm gonna let myself down. Watch the video. This was pretty good, but let's get into it, all right? So let's talk about making the proteins and how we're gonna get them to making these big shapes, right? Or like they're actually getting the job. So when we start out there, you can look at something like this. We have our 20 amino acids, right? When we're looking at them, for the most part, the big changes are gonna be coming from these R groups, right? So their behavior, how they're gonna behave is gonna be like the non-polar ones. I want you in your head to already be thinking hydrophobic, right? Those are gonna be setting up, if you enrich them in a certain part of your protein, they should behave and fold in a certain way. And that's gonna be key for the rest of these slides, right? Normally when a protein is being folded, if it's made to be in the cytoplasm and the nucleus, it's in a water environment, right? So your hydrophobic regions of a well-folded protein are normally hidden away, like you don't see them. If you don't fold the protein properly, it'll be sticking out, right? And then that's how your system's starting to recognize that something's wrong with your protein. So this is gonna be a key thing that'll bring up this whole non-polar hydrophobicity, right? On the other side of that, you have these other three groups. So these are all three polar, right? You have your regular polar at the bottom, and here you have two that actually are charged. You can either have a positive charge or a negative charge. This again is gonna be another feature that's gonna set up your interactions. Polar molecules like to interact with polar molecules, and then these kind of complement each other, right? Positive charge and the negative charge are likely to try to make a hydrogen bond. So these are basically things that help hide your amino acids away. On this side, these three, their interactions normally meant they give you like this really nice shape. And they also make like landing dots. So like sequences you can really recognize are amino acids. Right? And then the cool thing about this is like, because we have so many of them, at each position, you can potentially put any of these molecules in there, right? And because of that, you basically can make this crazy huge combination of what amino acids can be made at each spot. So 400 amino acids, potentially you can put 20 in each one of those spots. Depending on what you put, you can make these small neighborhoods of like hydrophobic pockets. You can make bigger things that like distal regions of the protein can come together and work together. Right. Well, let's go a little bit more into it. All right, so this is from the first part of the class. For peptide bonds, feel comfortable with that. And then here you have this dehydration reaction where you're basically losing water, and this is how we normally glue them together. Yeah.
You said we don't need to have any of the amino acids memorized, right?
No. On Friday or Wednesday, I'll finish making my, good question. I'll finish making my sheet. I normally always try to give you guys one in the test like this. I don't feel like doing those like translate this mRNA to this protein because I've done that before, like in another, I push them a lot. I'll push the table to be more something like this about how the amino acids and their charges. And the other one is, I don't know if you guys have been noticing, we've been getting specific amino acids can have certain modifications, right? The big ones is serine 309 tyrosine, that phosphate group, the sulfide bonds, and I think proline was another one they brought up. But I'll bring it up. I'll make my slide for this one first, like the nicer PowerPoint. And then in the test, I might just hide some of the keywords there. So for you guys to fill them in.
Okay.
But don't
And
memorize.
you said for the, when we do review, you're gonna do your like condensed notes.
I'm gonna do my condensed notes, yeah. If stuff in the condensed note doesn't make sense, right, that means that there's like, because when I make my condensed notes, it's normally like I'm reading through it and I'll be like something that I've memorized or something where I'm like, hey, I feel like everyone has a strong grasp on it. I take it off from my condensed note because they'll be like, hey, you're good on that. So if you read through my condensed note and they don't make sense to you, that means that you need to go back to those slides and get more background on it. Yeah. All right, let's go back. All right, so we're building chains, right? You can basically glue this reaction, right? It'll bring the carboxyl from one side, the amine group from the other, and you can glue this together. In today's lecture, we're going to be talking about how when the system goes bad or a protein goes bad and you want to start clearing it, getting rid of it, what we're going to do is we're going to do the opposite of this reaction, right? We're going to have catalytic enzymes that are going to grab our already made chains, and we're going to start breaking them back into these smaller like individual amino acids, right? That's our body's way of recycling proteins. So today when we're talking about there's going to be catalytic sites or catalytic enzymes, just know that I'm talking, we're going from the bottom, going back up. All right, so let's look at how a protein can be made. This one, I expect you guys to be comfortable with these, right? So it's basically 4 levels that you can have a protein organized. You have your primary structure, which is literally the order that the amino acids come out in. So for this one, you guys should feel comfortable with the N terminus and the C terminus. Right? So kind of like for DNA, remember we talked about 5' is the front, or like the head of the molecule, and then your 3' is your tail. For protein, your N terminus is your front, your C terminus is your back. All right, then you have the next level, it's going to be your secondary structure. For this one, they've only shown one up here, the alpha helix, but there's more types. The second one is called the beta sheet. You have beta sheet that looks something like this. And then you have the other one, which would be a random coil. So in this one, what's happening is for the secondary structure, you're looking at how the like distal parts, not distal, but like neighboring parts of the chain are basically interacting with each other. So here we're looking at the alpha helix. Each amino acid here is basically interacting with the one above it to help you make that move. In this case, it'll be this amino acids on this part of the chain interact with the ones from this side of the chain. And that's what they kind of link up to each other. In this one, it's a little bit, normally where you find the hydrophilic amino acids, and they interact more with water, and you just get this thing that's not really like a super strong structure. These, a lot of them, you can get them to, if you express them, even without any helpers, those are able to fold into that conformation. This you might get, like, you'll find intermediate, sometimes it's stable, sometimes it's not. All right, so that's our secondary, right? So you can update your notes here, alpha helix, beta sheet, and then random coils. Next level to that, it's gonna be your tertiary structure. And this is to be like, when you start making that bigger aspect of a protein, this will be things like your disulfide bonds, right? That means that you have this whole chain, and you can actually link up the cysteine. You see that sulfur with the hydrogen group? You actually remove those hydrogens, and those sulfurs will link up across a bigger part of the chain. But that'll be like a covalent bond, right? That'll be part of this one, the tertiary structure. Right, so your base chain, how the whole thing is going to be built up. And finally, we get to the quaternary, and it just means that how our protein interacts with other proteins. You know? So in all these cases, when you're building up, you're starting to build these two. The big goal there is you're making your protein to be like on a stable conformation. A big part of that stable conformation is you have to hide the hydrophobia. Right? You can either hide it by coding the amino acids into themselves, kind of like, in my head, I think like hydrophobic would be like our intestines, right? Our cells are hidden inside the body, our outside skin is our hydrophilics, right? If one of us had our intestines hanging out of our body, you know, it would like functionally, we would notice something's wrong with them, like an eye popped out or, you know, the gut's hanging out. Just like that with these proteins, hydrophobic region is sticking out. We've messed up the function, we know something's wrong with it, right? Some of them we can fix it in here, they can fold it in themselves. And some proteins, they get all the way to here, where it'll be like, they might have hydrophobic pockets, but those hydrophobic pockets normally interact with other hydrophobic pockets from a different peptide, and that's how they get stabilized, right, to make this aggregate. But that's the thing we're gonna keep an eye out, all right? So here I'm just showing off some really cool shapes that they can achieve. So you can see they can make like this little cylinder, almost like donuts, nucleoside transport. This is pretty cool. So this one, that you can see, it makes like these multiple beta sheets in a ring, and then it's basically going to end up making like a little tunnel you can put these through that domain, right? So in the plasma membrane, this is critical. Normally it blocks you from moving anything across. You put one of these pores, you can actually make a hole right through the membrane. You can get different molecules through it. Let me see. The cadherin, that one's pretty cool because it's like, it'll grab on to like the extracellular matrix of different cells and it actually brings them together. So it uses this half and this half to recognize the different like elements of the outer part of your cells, to actually grab them and kind of grip them into each other. Yeah, so they're like, they all have these cool shapes. They mix up these beta helix alpha sheets. They'll work with each other. You can also make them work in big complex, all specialized to get a 3D job done, right? Like in my head, I always think of this is like the hands of like our bodies, right? Super useful. All right, so let's think about the stuff that, again, the big thing I want you guys to focus here, and I'll bring up a lot, is gonna be that hydrophobic effect, right? Normally those are gonna be the ones that are gonna be hidden. Proteins do have other spots. Like I said, those ionic bonds, right? Where we have a positive and negative charge. If a protein has the opposite charge, it'll let you attract it. Hydrogen bonds, another one. If my protein spaces out my amino acids properly, I can set up like carboxyl groups or hydrogen groups to interact with another protein that might have their amino groups, right? Like those positive and negative charges, let us couple into each other. The van der Waals is going to be the weaker forces, right? And then for the covalent bonds, like I said, the big one, it's going to be this guy, the cysteine residues, right? Because we're going to be doing those sulfide bridges. This one, I'm not going to go that much into it, because it's a little annoying, but it's like, for the protein, it's a small side chain where it'll link back up into the, so the R group will react, I believe it's with the amine group in the front. What it does then is, if you guys remember from like OChem, the way these are set up, you call them cis or trans based on how your molecules are kind of popping out of that RNA chain. It ends up switching that. So what it does is if your amino acid chain was coming this way and it was balancing, kind of going to the front, it'll do a trans version like that, and now your molecule goes the other way. So but it's a little annoying. I feel like it's more OChem, so I'm not going to bring it up that much. So we'll put that one on the low pile. But the cysteines, please feel comfortable with that one. All right, so Zooming into that one, this is what it's gonna happen right here. You have your long chain, your SH, that's your R group, right, for your cysteine. And this is where we're gonna have this guy. Your PEI is the one that's gonna help. And what you're gonna end up doing is, you're gonna steal the hydrogen groups, and it's gonna help catalyze this reaction. Where you see we have now this covalent bond between these two sulfurs, part of these cysteines. When you do this, you're basically gluing these two chains together. Some cases, this helps stabilize it more. In other cases, this could be like the initial step to get it into the right conformation. Right? But the thing is, now you have this really strong bond in the middle of the protein, helping it to hold its shape. All right. Let's go back to this one. All right, so when proteins are getting made, there's gonna be things that normally are there to help. So this one we're going to cover with are gonna be chaperones. So again, imagine your chain, your amino acid chain is getting made. But let's say there's certain parts of this. Let's say this little beginning part maybe has like hydrophobic regions that interact with something that comes out here at the end of the chain, right? While you're making it, if it starts making it and you only let the protein out, that hydrophobic region is going to show up and be in water. If you don't have chaperones, only that region would be like super unstable, right? It'd be also getting targeted for degradation because we have our like safety mechanisms target for hydrophobic regions to look for bad proteins. So if you have these proteins called chaperones, they can temporarily kind of hold on to that, and they'll serve to stabilize the protein when it's getting made. Once you build the chain long enough that you can now it can go into a stable conformation, then you can release the chaperones and it gives it time for it to fold into itself. Right? We're going to go over one of the heat shock proteins, which kind of helps with this. And what you'll see is that they also invest energy themselves, the chaperones, to kind of help with the protein folding. So we're going to get into that one right now. But before we go there, if everything goes well, the chaperones give you time to make the whole protein so it can stick to itself. If the protein isn't properly folded, I believe the chaperones can reversibly bind back to it, and it gives you more time to try to get the folding to work properly. If they don't help and you can't get the system to work, what's going to happen is this protein might be improperly folded. You'll have hydrophobic regions sticking out. And then what's going to happen is multiple proteins, like any other protein that's misfolded, might have a hydrophobic pocket sticking out. At that point, those proteins will start clumping up with each other just to be in a safe environment, right? So the more bad proteins you make, the more of an environment you have where they clump into each other and it gets harder to try to pull them apart. Yeah. That's where they're big players. So here, heat shock protein is one of these big chaperones that's going to do the job. This one's actually pretty nice and pretty fancy. What it does is it traps that protein, brings it into one of these little binding pockets, right? And then it's going to use this cap. For this one, I put it in here. So, so it's two parts, right? You have like a little cup that's holding your protein. You also have a cap, a little red thing at the top, right? With this one, if you go into the details about how it works, is that cap is going to help us recognize the unfolded proteins, right? Then this little cup is basically going to get sealed up, and inside of that, you're going to make a microenvironment to give you yourself a better shot to refold that protein. So inside of that, you're going to be giving that protein basically time to try to refold itself. At which point, if everything works out well, you then can dump out the properly folded protein. If not, you're going to end up having to tag that protein to basically throw it away. Right? So this is one way you kind of give the system a shot to try to salvage what you already invested in. If not, you're going to dump it out. I have my notes here. I remember I was thinking about the cap. I believe the cap gets recognizes ubiquitin. But it should be, it's not here, I'll show it later in my slides. Again, the big answer for this one, how do chaperones recognize misfolded proteins? It's going to be the hydrophobic sticking out. That's the big clue. All right, so here we go. this is one of the mechanisms that we can tag a misfolded protein, and it's gonna be coming from, let me see, so it's called the ubiquitin proteasome pathway. Ubiquitin is gonna be the tag that we use, right? For this one, it's the E1 gets activated, or it activates the ubiquitin, right? It then basically gets transferred to E2. E2 is gonna hold on to the ubiquitin. E3 is gonna be your intermediate. So that one is gonna have the ability to recognize E2. At the same time, it'll also recognize to your target protein. So it means if your protein's misfolded, throwing some hydrophobic pocket, E3 is the one that's actually gonna be able to recognize or target that misfolded protein. Once you do that, these two things come together, and then your E2 will transfer that tag onto your protein of interest, right? Now, this process, it happens over and over. And like I said before, if you get one or two tags, like I've seen, it just slows down the protein's function. It gives a little bit more time to fold. But once you build up too many of these tags, it kind of like gets built up on top of each other. At that point, you send it here to the proteasome, right? So this is the thing we were talking about before, where this is the one that has a cap. That cap recognizes these ubiquitin tags. That's what helps you bring in that protein to this part. Inside of this, which is the little body of it, for this one, it has catalytic enzymes on the inside. And this is where it's going to be cutting the amino acids, right? Like I was talking in the beginning, you basically add water, and it'll break the amino acid chain, two different amino acids. So at this point, this is how you can degrade this. You are going to use ATP to kind of push things through, but the main thing is going to be happening through enzymes here in the middle, just chopping up your proteins. That make sense? Yeah. All right. So those are the big goals of how we're going to clear stuff out. Yes.
Are we gonna have to know like the differences in function between E1, E2, and E3 for the
1, 2, 3.
test?
1, 2, 3. Let me think. There, E1 doesn't give me, like, to me, E1 is not that interesting, right? It's like an initial step of charging up the molecule. E2 is another one where I'm like, it's kind of interesting in the sense that it's going to be the one to transfer the molecule in. But if you guys look at E2, I believe it says that there's only 20 versions of these pathways. Sorry, E2, there's only 40 of them, right? So it means it's like a general mechanism. The cool thing that I really like about this is just this, that E3 has about 600 different types. Right? So having that big number, and then the fact that they have that dual job where they can grab onto the broken protein and they can grab onto E2. Right, so that's the big mechanism of how does ubiquitin regulate what proteins to degrade, and it's like we have a lot of specialized E3s that can recognize misfolded proteins. So E3 is my favorite. E2 and E1, I probably wouldn't talk about them that much. Yeah.
Like, please, can you go to your slide?
Say it again?
Can you go to the previous slide?
Yeah.
Okay. What's the difference between these two pathways that you drew? Like, are they like continuing each other?
Yeah. So sorry. Yeah. So this one, this arrow, guys, here right here. It's the same picture. So this guy. What's going to happen is this is going to keep getting sent to getting more E2. You need more E1s to get the energy to the E2. The E2 then needs to do it one more time to keep adding more ubiquitin.
Okay, wait. So basically, what happens is that the E1 recognizes the protein.
So E1 up here,
Yeah.
it recognizes ubiquitin.
Okay. Yeah.
So that's a little green tag.
Okay.
We charge up that reaction, or we catalyze the reaction of getting the ubiquitin to stick to E1 by using ATP energy. That powers up the reaction, right? Now that we have, like, let's say, like this high energy bond, we can transfer ubiquitin to E2, and now it's holding the ubiquitin tag, right? And then this is where the hard worker comes in, not the hard worker, sorry, the specialized worker comes in, right? The E3, it'll recognize the protein that's misfolded.
Yeah.
And you basically have a library of them expressed. And whatever this protein got misfolded, it'll recognize its own E3. Then you have your E2 will transfer the ubiquitin tag to the protein of interest. Yeah. Yeah.
All right.
Why does it keep getting more times?
I think it's a timer to give them time to get them fixed.
Okay.
Right. So it's like it'll tag them if your protein, let's say, has the ability to refold itself, and it can do it. It's no longer going to be recognized by E3 these ubiquitin tags are reversible, right? And you'll get rid of it. If your protein, they tag it and it's still not getting fixed, it's gonna make this pathway happen again. And the longer you're staying as a badly folded protein, the more times you go through this pathway, leading to that buildup of this. Now that you have a crazy amount of ubiquitin tags, that means that they gave you a lot of time to fix it, but you didn't do it, so now you're gonna get it taken out.
Is E1 only like done once to recognize it and then it just goes between E2 and E3 to repeat it? Or does it ever, so like E1 recognizes the protein, but then as it's adding more, does it just stay between E2 and E3 or does E1 like have to recognize it again?
So E1 doesn't recognize the protein of interest. E3 is the one who does it.
Okay.
E1, like I said, that's why I don't like E1 that much, because it's just charging up the reaction, right? So it's getting the ubiquitin to bind here, right? Then it's transferring to E2. But even E2 isn't recognizing our protein of interest with E1. E2 just clamps onto E3. So the big player, that's why to me, big player is the E3 one, right? It can recognize the protein you want to mess up. It brings in the E2 to help you transfer that ubiquitin. But E1, I think you just keep making it. You basically have a pool of E1, and you have a pool of E2. The E3s are going around searching for the target proteins, and then it'll transfer it to whatever target it found. Yeah. All right. Cool. All right. So now we're going to get into the mechanism of how does the cell know, or how does the system know what's a bad protein and how does it respond to it, right? So this one, I want you to think of 3 things like, how do we detect that there's a broken protein? I want you to think about what kind of response we have, and then I want you to think about if we produce any kind of signals to regulate. Right? So as I go through these today, break them up into these 3 steps. This is where I really think the midterm stuff's gonna come up. Right? This first one, I talked about it last week, which is the ER-associated degradation. Right? So in this one, the big background to it was, when your protein's getting made, dumped into the ER lumen, you're gonna get sugar molecules added to it, right? I think in the yeah. So it's gonna be my first thing out here, right? This ERAD program, ER-associated degradation. In the book, it says they're gonna get N-acetylglucosamine, B, you're gonna get mannose, and you're gonna get glucose. So you're gonna get 3 sugar molecules. Here they show it's like purple, light dark purple, light purple reds. The only ones that I really care about are gonna be the glucose at the end. Right? So what you're gonna notice is, here's our protein, it's got modified, there's your sugar molecule. What's gonna happen here is, this calreticulin is gonna use that protein shape and your sugar molecules to basically get it into the right, kind of like guide it to get it to the right conformation, and it's gonna be cleaving that sugar, right? Once it gets pushed out, now you have your sugars released without any glucose. If the protein is properly folded, right, AKA no hydrophobic regions are shown, that protein can use this sugar tag to send it to the next vesicle. And so the vesicle says, right, you keep going on the pathway. If that protein has hydrophobic regions sticking out, right, stuff that it shouldn't have, that's where this big player comes in, this UUGT, right? UUGT is gonna give us that first job, which is gonna be detection, right? UUGT recognizes hydrophobic regions on your misfolded proteins. So that'll be step 1, right? It'll be this big guy, UUGT. UGGT, UGGT, hydrophobic binding with proteins. So that's our way to know that something's wrong with this. You have hydrophobic part sticking out. The next thing that it's going to do, the GT part, is it's a glucose transferase. That means it's going to add a sugar molecule. Again, the big key player is that glucose. The one that we heated out before, it's going to attach it one more time back to it. And it's going to, once it has that sugar molecule, that glucose in it, it can go back into this calreticulin. I'm going to call it cow. It goes back to cow, and then cow, you have one more chance that this protein's going to cleave that sugar, but it's going to give that opportunity to refold the protein properly. So here we have like this soft-handed approach of let's fix this protein, right? If it didn't fold properly, hydrophobic's leaking out, we'll add a sugar to it, we'll go back into the concentrate to refold it, and then we'll send it back out, give it one more shot. On the other side of that, if it tries this and the protein is not, it's still getting misfolded, then here you have the alternative pathway going this way, right? Which at this point, you're gonna start removing the nano sugars. So it'll be the next level up. Before we just worked with the glucose, now we can start hitting these sugars up here. Those sugars are basically gonna be the tag of if a protein's been in that environment for a short amount of time, the sugar should be present. The longer it's been in that environment, the more they're gonna get eaten away, right? And again, this will be the long-term response where like, we gave this protein a shot, it didn't wanna do what we said, so now we're gonna have to get rid of it from the system. And that's where this is gonna happen, right? We're gonna help remove those sugars. Here, just remember this retrotranslocation is basically gonna kick this protein out. Once it's in the cytoplasm, we're gonna have a mechanism to proteasome that they're targeted and degrade that protein. Right, so either you go the easy way and get it yourself straight, or we'll destroy it. Those are the two mechanisms to follow. Yes,
So the retrotranslocation recognizes that it doesn't have the sugars and then sends it out?
and kicks it out. Yeah, in my notes, it literally says calcitron is the folder, UGGT, that binds hydrophobic. It's a sugar donor, so it'll be like your fixer, and then the retrotranslocation is the one that kicks them out. Yes, sir.
Okay, the UGGT only recognizes the hydrophobic part of the protein that's outside the protein.
Yeah.
Oh, okay.
So for example, let's say we were, the proteins themselves, I'm UGGT, and let's say you're the protein of interest. If I see your guts hanging out, I'd be like, oh wait, hydrophobic, like his guts are, and I'd send you somewhere.
Oh, okay.
It's like that.
So the cal,
Yeah,
so that is how all proteins are usually folded, or that's how like some proteins are folded?
that's what some proteins are folded, like it helps out, and it has recognitions of the sugar and of the protein of interest.
And the sugars are put on whenever it enters the ER.
Yeah, so I think last slides I had them. I didn't go that much into them. But like I said, the big player there is that glucose is that really big signal of like, if you folded it properly, if you already went through this cal system, you get rid of the glucose and you give it a shot. If it's busted, this UGT will recognize it, give it one more sugar back, and then try it one more time to refold it.
So the sugars are added to all proteins?
When they're coming in, yeah.
Yep.
So this is, we're talking specifically about proteins coming into the ER, and this is where they're getting glycosylated as they're coming in. So yeah. So we do cytoplasmic ones, we have to look them up. Right. But the focus here is that glucose is just the big signal of like, either we're gonna fix this, and we got a shot to redo it, right, which is this way, you can cycle through this, or if we have no hope, we're gonna get rid of even more sugars and we'll dump it out with this retrotranslocation. You don't remember the names, you give me the task, and I'll be okay in the midterm. Yeah. All right, just how we have this one. Now we have this thing called the unfolded protein response, UPR. Another one, I'm thinking, the question how they're going to be compared these two or compare these with each other, but all these have really cool mechanisms of making sure that the system's okay. Right? So first, let's go into this one. It's called IRE1. And basically, it's you have a protein that's transmembrane. Kind of like showing a little picture. So basically, it's inside in the ER in the lumen. It goes through the membrane, and now you have this outer portion, right? It's going to be all the way to the cytosol. In this case, this upper part, the one sitting in the ER lumen, that's basically going to be this first thing. How are we going to detect that the protein's misfolded? Any guesses on what it has on the inside? How do we know if the protein is folded?
Hydrophobic,
So what do you think it has on the inside?
hydrolytic.
So it has hydrophobic pockets and it's just waiting, right? So here, this would be the little drawing here. This would be the inner part, hydrophobic, right? So it's looking for a partner. They're normally built up. They're already sitting on the plasma membrane. So they're already there waiting, kind of like, is something gonna go wrong? when nothing's wrong and they're by themselves, they basically, they have these two jobs. One is they can be a kinase, aka they phosphorylate other proteins. The other part is they're also ribonucleases, aka they can cut RNA, right? If there's no unfolded proteins, they'll be calm, they won't do anything. The moment the unfolded proteins, like those hydrophobic pockets, bind on the inside, these two complexes start to clump up to each other, right? So the detection of the misfold is that hydrophobic stuff. And the first part of the response is you're gonna have these things that become oligomers, right? So multiple of them start coming up together. The other one is they start becoming kinases. That means they start adding phosphate groups to each other. Those phosphate groups act as activating marks, which makes their endonuclease activity, the ability to cut RNA, it becomes super efficient, right? So now they're outside cutting RNA. Now here's where we're gonna get to the signaling part, right? So again, we have a drawing, they're getting activated, they're gonna cut this RNA. So at this level, we're gonna be looking at this factor called XBP1. That's the big thing. This is basically a transcription factor, right? AKA it binds DNA, it can turn genes on. When the system it's activated, there's a specific intron that gets cut out. When that intron gets cut out, these exons come together and they produce the actual functional version of this. Right? So this XBP1 only gets made when the RNA gets properly spliced. That then is able to move back into the nucleus, and here we're going to have gene regulation. Right? For this one, if I remember correctly, it's going to turn on the expression of chaperones, right? Remember the things that help us fold proteins? It's going to turn on chaperones. It's going to send signals to try to activate this pathway. It'll try to activate the EUM program. Right? It'll be like, hey, let's try to see if we can get these proteins to get fixed. And it'll activate other genes involved back into the system. Like it'll make this thing get turned on even higher. Right? So it's like if there's misfolded proteins, we send the signal back. Yeah. All right. So cool. So this is why I really like this for the midterm, right? Because we're getting here. It's like how proteins working with each other based on this interaction. We can get this phosphorylation and this DNA cutting up RNA cutting ability to show up. Right. Here, this is super cool because you have a pre-mRNA that if the system is turned off, everything's okay with your cells. You're making an mRNA with an intron that basically doesn't work. It doesn't do any job. The moment you need it, you can splice it, and now you make a functional transcription factor down here. And then this is the one that regulates the genes that you care about. So it'll be like, hey, turn things on to help us out. Yeah.
2 questions. One, do we need to know about the functions before it sees an unfolded protein?
What do you think? I just know that it's like, I won't say yes, just because the whole point of the system is like, none of these things are ready to go, but they're not doing anything until the moment we need them, right? Kind of like this, it's like, your proteins are there, these, like EIRE1 is present, but it's literally, because it doesn't have a phosphate group, it's not doing anything, right? It's on its constant. The moment you activate it, now all of a sudden it wants to be a kinase, it starts eating phosphates, other stuff, and it starts cutting RNA. That's how that works. Even the RNA thing, I can appreciate it that sense that like that gene has to be getting made, it has to be getting transcribed. Now we have mRNA being made. But because it has that yellow gene that's not the function, it doesn't do anything. Up until you need it, now we splice it, and now it's the active version.
How is this different than a spliceosome? Is it just if the spliceosome doesn't see it? Like the endonuclease part of it?
So remember that they use targets to help find them. Like spliceosomes don't randomly cut like RNAs to make them. They normally get guided to pick your specific target. I bet you this thing, when we go look at the papers, like the actual like the primary literature for it, I bet you they have guides that help it find exactly where to cut here and where to cut there to make this happen. I just don't have the picture.
So it performs like a similar function.
Exactly.
And then after it splices and puts the 2 exons together, the signal just tells it to, what does the signal do now?
So that's at that point you have an mRNA, right? right? You have one new mRNA. That mRNA has to go through translation to become a protein, that'll be the transcription factor. And then once we get down here, that transcription factor is the one that can go into the nucleus, bind the promoter of the genes, and turn the other genes on. And that'll be it'll get us more chaperones, it'll turn this on, it'll turn that back on.
So it tells it to get translated.
Yeah, it'll be, sorry. The mRNA gets modified, then it still has to go it has to go translation to become a protein. Then as a protein, it can go work on multiple genes, and it'll turn their transcription on. So they'll start making RNA. And then they have to go through a pathway of becoming proteins, and then they go through
What genes are being activated? in the DNA?
Key ones for this one are chaperones, genes involving chaperones, for the chaperones, the UPR program, and then this UPR. So it literally goes and activates more of these genes. Right? You can imagine if your proteins are folded properly, you get more of these, the cow protein or the UGGT, now you have a better chance of trying to fix it. How does this one feel? It's pretty good about it? Yeah? All right, let me go the other two. I'm not gonna go in as great detail as I did with these drawings, but I'll talk you kind of through them. They all have like a really fancy mechanism of sensing it, modifying it, and trying to send out a signal. Yeah, my bad, I should have jumped to the next slide. This guy. All right, so for this one, there's another one called PERT. There's another one that's gonna get activated. It's gonna work the same way. It's gonna be sitting, it's a transmembrane protein, half of it's gonna be inside the ER, and it's gonna be, again, these hydrophobic pockets, waiting for this folded protein to show up. Before it shows up, it's not gonna do anything. The moment it shows up and the protein binds, all of a sudden, that kinase activity gets activated because they clump into each other, so they phosphorylate each other. Now they're on the on state, it's time to get their job done. The cool thing about this is that what they phosphorylate is BIF2. So you guys remember from earlier before, eukaryotic initiation factor 2. All I remember that you need to remember for those is we're talking about translation. When it phosphorylates BIF2, it inhibits translation. Right. So yeah, so my notes I put it's an oligomer, it's a kinase, and we're gonna stop the function of BIF2. In that sense, it kind of makes sense, right? You have a bunch of misfolded proteins. The last thing you want to do is keep making more proteins that are going to clump up with your stuff. So what you're going to do is you're going to stop protein production. That way you give yourself time to fix things up. Yeah. So that's one of the big roles that I like about this one. The other one that it does is it activates ATF4. It's another transcription factor, right? And again, this transcription factor goes and binds the promoter of genes that are involved in the UPR. So it's a self-activating system. You have too many misfolded proteins, you activate this, you turn on translation, at the same time you turn on this transcription factor. The really cool thing, which also again is super cool, is that this transcription factor can get expressed despite you trying to block off the EIF2. So it'll stop making other transcription factors, like other proteins, but it keeps making this one, preferentially. So you keep getting more ATF4. AKA you signal back to tell it to make more UPR. So it'll make more of these signaling proteins. Makes sense? See, this one I like it because it connects back to like translation. When you bring up the EIF2, it's a really cool mechanism. Like I said, it's like if in your cells you have something that's going bad, you're gonna have your chaperones busy trying to fold these proteins, right? You're gonna have your cow, your UGT busy trying to get these things to work. If you keep making new proteins, those proteins are not gonna have any chaperones. They're not gonna have any of these folders. Automatically, it's gonna make you more and more bad proteins. Right? So it'll be like a bigger pile of cars. So first thing we'll do is stop traffic. So super radical power for this one. All right, next one, we're gonna see the ATF6. This one is a little bit, it feels relatively boring compared to the rest, right? So for this one, again, it has an internal part like this. The internal part normally binds to a protein called BIP. BIP. But this protein, it has affinity for the misfolded protein. So in case they start building up in the environment, your BIP protein, instead of binding to this factor, will bind to the broken protein or the unfolded protein, In that case, that relieves the signaling coming from the ATF itself. Normally, this helps keep it inside of the UPR, it being interacting with the bit. The moment you take this out, the ATF6 moves to the Golgi apparatus. Right? In the Golgi, what you're gonna find is that there's enzymes that cut the protein. And what they're gonna do is they're like decapitate it. They'll cut it at that transmembrane domain, and they'll release this part. Now, this part that they release, that's actually a transcription factor. So again, we get to this level here. So we have ATF6 would be that active transcription factor. Surprise, surprise, right? It's gonna go find the genes that are involved and regulating the UPR. So it'll be another positive way of like, if something goes wrong in the system, it signals to itself to make more of the repair mechanisms, like turn them off to the higher levels. All right. So for all of these, I'm thinking this one, I like it a lot, right? Because it does this whole quick repair mechanism. We give the protein a chance to fold, right? Everyone feeling cool about this one? The ERAD? Yeah. All right. For the UPR, here, I like that there's, all of them are trying to find ways to fix it, right? They have a way to detect it, right? Some way to detect that this protein. They have a response. I like this one because it goes from, it talks about splicing, right? So that's another cool thing we can talk about. We can compare it to this one, where this one talks about there's really translation control, right? Or there's talks translation. And that's the way that we can get the system to fix it. While this one is, we're gonna make more molecules that are trying to repair the system. So there's still in a repair mode. This one also works same thing. I think if you feel comfortable, maybe one or two of these. You don't have to memorize all three. Just understand the general thing. I might ask you a compare and contrast type of question. Yeah, especially about like, I think this step is really important to me. Because it really brings all the stuff that we talked about together. The kinase activities, these interactions, splicing, basically the code of how we can block out translation. If you bring up those in a thing, I'll be fine. I'll give some really good points for that section. So again, this slide, feel comfortable with it? Here's the same slide. They did it with a little bit of different drawings, so sometimes this kind of helps my memory. You can see here kind of what we're talking about, right? The first pathway. This is the long version without splicing, aka this is not a good transcription factor, it doesn't do the job. The moment we cut it out, now we can make the right protein, and we can go send it out to get the right genes, right? And the next case is, once you have a misfolded protein, you can activate it, they kinase each other, we phosphorylate the uric initiation factor 2, aka we break it, we stop translation. The ATF4 doesn't care about this, it'll still be getting translated, and that's the one that's gonna go lose the signal, breaking the genes. Or in this case, which is the ATF6, BIP, it's stolen away to these unfolded proteins, then it's allowed to move to the Golgi. Here, you're gonna get 3. Now that's a transcription factor, and we'll get it to work. Yeah? All right. In all cases, we're looking at different ways things are getting modified, right? Splicing, phosphorylation, and cleavage of proteins to give us a signal to get this thing to work. That's why I was like, this is a really cool slide. All right. I think that's it for today. Any questions? Good? All right, perfect. You guys,